Barry, great to have you here. Really looking forward to this discussion. For those of you who don't know Barry, he's a top innovator in synthetic biology. You've had various roles spanning from academia to large biotech, and you founded two deep tech companies as well. You have ran multinational genome sequencing efforts. You develop novel sequencing, chip technologies, and you have more than 50 patents as well. I've had the opportunity to work with you at Human Longevity, and it was a very transformative experience for me personally. You're also the advisor of our fund, Nordic Science Investment, and very much looking forward to this discussion on synthetic biology. Let's kick it off with your company, Avery. Could you tell us a little bit about the vision of the company?
Or, maybe I could give a little bit more background about myself too, that I think frames
Yeah,
a little bit of how I approach things.
yeah.
Because I consider myself to be fundamentally a technologist, and I was a professor at UCLA for 20 years. But even there, it was my whole goal to try and what do technologists do? You try and use technology to solve really big, important problems. And so I'm actually a mathematician by PhD. I was a professor of math and then physics and then nuclear engineering. And my whole past life was about energy. So what's the big problem? Well, energy is always the biggest problem. And so I had a past life working on energy research. And what is the ultimate solution to energy from a technological perspective? It's nuclear fusion. It's how the sun works, right? And so I had a whole past life designing nuclear fusion reactors. And I did that. And in fact, I worked on the design team of the largest reactor that's ever been designed. It's called ITER or ITER. It's being built outside of Marseille, France right now. And it was a project that was actually launched by Ronald Reagan in 1986 in a State of the Union speech. Because it was such a big effort to develop fusion, Reagan proposed that the four historical enemies should come together to do it, namely the US, Soviet Union, Germany, and Japan, to unite their efforts for the sort of the good of mankind. And so that was a massive project. I ultimately was on the design team for that. And when we finished that design work in the early 90s, to me, it was clear it would take 30 or 40 years to build this machine. Fusion reactors are the most complex machines humans have ever devised. And, it was going to cost $10 billion. And there was only going to be one of them, and they didn't even know which country to put it in. So it was clear to me, and I told all my colleagues, like, it's going to take 30 years to build this. And they're like, no, we'll break ground next year. I'm like, no, it's going to be 30 years. And so rather than sit around for 30 years waiting, and I was right, by the way, it's near completion in Marseille, France right now, and the cost has ballooned to $40 billion. And, I was sort of right about all that, but it's still very important. And, I'll go back to it. When they turn that reactor on in another 5 or 10 years, I'll go back to working on fusion. But, if you ask yourself, what are the other big problems that could be addressed with technology that really impacts society and mankind? They're all biological in nature. All the other big stuff aside from energy is biological in nature. We need to cure all disease, of course. And we also just need to learn how to engineer biology to do all the other things we do. And so, to me, those were the two big goals, and where we have to develop new technologies. And so I've had to shift my focus to that. And I didn't know any biology at all, like zero. So being a professor at UCLA, I took a sabbatical ended up being a 2-year sabbatical to learn biology firsthand. And so I actually went and worked in a molecular biology lab. And, this was at the time when the Human Genome Project was just becoming a thing. It was near completion. And so genomics as a field was quite, active. And of course, that was the whole premise behind precision medicine. The genomic techniques are going to let us develop precision medicine. And so curing all disease, precision medicine, that's a big part of it. And so I decided to focus there. And, as the human genome became sequenced around that time, I completed my sabbatical and wound up sort of running a core facility at UCLA. There was actually a national level genomics core facility. PIs from all over the country and even internationally could do their genomics projects with us. We were sort of like a mini genome center. And so we had all the state-of-the-art genomics technologies. Back in the early days, these were DNA microarrays, but it was all turning into next-generation sequencing. We were like an alpha test site for all the first of the next-generation sequencers. And we did some very big early sequencing things. My group sequenced the first cancer genome that was ever sequenced. We did the first epigenetic sequencing of a large genome that was ever sequenced. We did the methylation sequencing of mustard plant, Arabidopsis. And so we were doing these big genomic things and working with all the state-of-the-art, emerging sequencing technologies. And it was clear that that's how we were going to impact medicine, and health, what was using sequencing knowledge to do precision medicine. And so because of that, I got invited by what at the time was the leader in sequencing technology. Applied Biosystems was the historical leader in sequencing. They made the first commercial sequencers in history in the 80s. Their commercial sequencers powered the Human Genome Project. And so they obviously wanted to maintain their leadership. So they actually invited me to leave academia and come lead sequencing technology development at Applied Biosystems. And, I said, no, I don't want to do that. We're doing cool things here in academia. We're sequencing the first cancer genome. We're sequencing the first epigenome. We're developing the foundations for precision medicine. But they said, look, if you come lead our technology development efforts and you find like the next powerful sequencing technology, we'll put $100 million into, helping you develop it and bring it to market. So that changed my thinking. And so I said, look, I will come if we can do a Manhattan-style project to develop sort of the ultimate sequencer. And to me, the problem with sequencers of that time from a technology perspective was they were very complex, costly technologies. The goal was $1,000 genome, but it was still like $1 million genome at the time. So you're talking about the technological question of how do we get to $1,000 genome? It needs to get 1,000 times cheaper, maybe 10,000 times cheaper. And as a technologist, to me, it was simple. What you have to do, what technologies ever make that kind of cost reduction? Just among all technologies, like you think of how we make things, what has ever gotten 10,000 times cheaper? And it's chip technologies. That's the only thing that's ever caused that kind of reduction in costs is putting things on chip. And so basically, my premise was to them, I'll come if the goal can be to find a way to put the reading of DNA on a chip device. And so they agreed. And so long story short, I rapidly left academia. We did do a Manhattan project. And the outcome of that was this. This is commercially is known as the ion torrent sequencing platform. These days, it's Thermo Fisher's sequencing platform because Thermo Fisher ultimately acquired Applied Biosystems. But the point about this device is that it reads DNA, but it's an all semiconductor chip device. It's all electronic, all transistor based. It's the same kind of chip that's in your cell phones. It just is open so that we can squirt some DNA solution onto it. But it's made by the same foundries that make the chips in your cell phones, and that's why it's so powerful and scalable. And that technology did scale to this. This first one had 1 million DNA reading sites, scaled to 1 billion DNA reading sites. And this is the one that could indeed achieve a $1,000 genome and beyond. And we did actually beat the world to the $1,000 genome with that. But that's why I left academia, was really to go from doing like foundational creative work to making impact in the world. And impact in the world for a technologist has to be making scalable technologies. And so if you think as a technologist about all of biology, you have a simple mindset. What's the foundation of biology is DNA. And what's important about DNA is two things. I'm reading it or I'm writing it. And so this first foray, was making the reading of DNA scalable and much cheaper because of that. And so, I knew at that time that the next big thing would be to take the writing of DNA and make writing DNA, cheaper and scalable. And again, the only way I know to make things cheap and scalable is to put them on chip devices. And so, conceptually, my next goal after the ion torrent chip, as far as technology goes, was to find a way to put the writing of DNA on some kind of chip, a chip that synthesizes DNA. And so that's where the company Avery ultimately came from. So my current company, Avery, its mission is to make it possible to engineer biology routinely. That's sort of the big vision. Let's just make it so that biology is engineerable. But the way it does that is by putting the things you do to engineer biology onto chip devices. Let's make it radically more cost effective to make and test DNA, because that's sort of at the heart of engineering biology. I got to make some DNA, I got to test what it does. And so, the mission is to make biology engineerable. The technological way to do that is to put the key things about writing DNA and testing it onto chip devices.
There's of course a lot of discussion now about synthetic biology. In your opinion, what is it that makes synthetic biology so interesting right now?
Well, synthetic biology as an idea has been out there for decades, maybe really going back to the late 70s when we first figured out how to make a gene, right? The origins of synthetic biology are we could make a gene, right? And put it into something. And so to me, what synthetic biology is in its most primitive sense is making organisms that do what you want. And that comes from making the DNA and putting it into some sort of organism where it can be expressed and having it do what you want. And so synthetic biology, to me, I keep it very simple. I want to be able to make an organism do what I want, and I want to do that by making DNA and testing it in organisms. And I got to do a lot of that testing, and so I think, to me, mentally, the inflection point in synthetic biology and the excitement was when Craig Venter in 2011 made the first entirely synthetic living cell. That's sort of the, not just a sort of transformational point in synthetic biology. That's a transformational point in human history. We could actually make a synthetic living thing. This was the goal of mankind going back to since recorded history. All of humanity's imagined making living matter somehow. And so Craig Venter finally did it in 2011. So to me, that's sort of very transformational. Before that, you made some genes and did some things. But then at that moment in time, it made it clear that you can go beyond genes and make entire genomes synthetically. And so, that's the excitement. Now, you might ask yourself, well, okay, why didn't that immediately just catalyze a world of synthetic biology? And it's because it's so expensive. What Craig Venter did took him a decade and $40 million worth of industrial synthetic biology to make that genome for a living cell. And so while it's sort of like going to the moon, like Craig went to the moon, but no one else can because it's so expensive and so complicated. And so until we solve that problem, we're not going to have a burgeoning, vibrant, impactful field. And it's sort of very analogous to like going to the moon. I mean, we went to the moon, but that didn't catalyze like some huge era of space activity. But now companies like SpaceX are sort of catalyzing a lot of space activity because they're making it affordable and practical to do it. And so I think that's what's missing in synthetic biology. The science is proven now. It's all about how can we make it practical and affordable to actually do things in synthetic biology.
So that's a very good point. And what do you think are some of the key elements for us to really unlock and for synthetic biology to shape society to its full potential? What do you think needs to happen?
It's very simple in my mind, because again, we've seen the full potential from making genes that do useful things, like a gene for an antibody that becomes a therapeutic, or engineering a protein that's useful, like some industrial enzyme that, makes your clothing softer. And so, we've gone from, yes, you can make genes and engineer the function of those genes for useful products, to making a whole genome like Craig did. And so I could make genomes and try and engineer on the genome organismal scale. And so what's missing is very clear, is just to make that economically practical. And so, engineering biology is fundamentally trial and error. We don't know enough to just design the DNA that does what we want, right? And so it's always trial and error based. You can have good guesses for things, but still, you got to guess a DNA sequence and make it and test it and do a whole lot of that and iterate on that as well. And so to me, what it takes to unlock this whole field is making that economically practical to do at scale. And so right now, as a practical matter, to make a gene and test it costs a few $100s. And because it's trial and error based engineering, I don't need to try just a few things. I need to try millions of things. And so, that doesn't work. If every trial costs a few $100s, I can't do millions of things. And so we need, in my mind, to make it a million-fold cheaper to do the basic process of making the DNA of interest and testing it. And it also has to be scalable. I mean, I have to be able to do millions of those as well.
Yeah.
So that, to me, is the heart of what will unlock the field.
And sort of from an investor perspective, we think about deep tech. Yes, it's capital intensive, patient capital, takes a long time. Do you see the same for synthetic biology, that it is capital intensive, or what? How do you?
I see it.
Yeah.
For synthetic biology, the way deep tech is going to work and the capital requirements are going to mirror what happened in the, let's say, the semiconductor chip world. It was very expensive technology to be able to get to make chips, right? The Intels of the world that created the ability to make chips and make processors, that required very large amounts of capital and patience. But once that computing hardware was available, the software world came and developed very rapidly, and it was not expensive to develop software. The same thing will happen in synthetic biology. We need some very difficult to develop, costly, sort of hardware advances, which is what I'm working on. But then once you have those, the rest of synthetic biology can become much more like software and can move very quickly and without a lot of capital. And so I view what has to happen is we have to get that sort of hardware infrastructure in place, which is costly and takes a long time, very similar to how the semiconductor industry did it, to unlock this software world, which then can be very dynamic and not have huge capital requirements. So I think the same thing will happen in synthetic biology. And if you look at, okay, what is this hardware foundation for synthetic biology? Right now, robotics is state of the art. If you go into the most sophisticated synthetic biology labs in the world, which like Ginkgo Bioworks is well known as a company, big pharma tends to have these for making biologics, it's robotic automation. And that is state of the art. And, if you could build a 1 million robot factory, you could do a lot, but a 1 million robot factory is not possible. And so, and it's the same with like the early chip industry, like early computing. When computers were made of vacuum tubes, it was just not possible to make computing infrastructure to unlock the software world. It's only when computing went on chip, and the scaling of that you could get the hardware infrastructure where it needed to be, low cost and scalable to unlock the software world. And so I think it'll be very much the same in synthetic biology. Robotics just doesn't cut it. We have to get to lab on chip type devices that do the experimental work of making and testing DNA, and that will unlock this world of synbio is now like software.
And how far do you see us being from that, from really unlocking and entering into sort of an explosion on the software side?
At this point in time, it's an engineering effort. In other words, we don't need any new technological advances. It's just engineering technologies that are already proven. And I'm working on that. Other folks are working on various aspects of it too. But that's the good thing is there's not like a scientific advance needed to make sort of the lab on chip devices that can really unlock this era. So, I view it as something that can be done in less than 5 years, frankly. And then you'll see a very rapid emergence of synbio apps,
Yeah. And thinking now, sort of, you're a scientist by heart, and then you've made sort of a very interesting journey into deep tech entrepreneurship. But do you feel that you're approaching entrepreneurship in a different way, thinking of sort of being a scientist at heart, and how do you see that entrepreneurship being sort of a former professor at the
Yeah,
university?
I do, and I approach it as a technologist. Like my contribution is going to be, conceiving of and creating these technologies that are powerful. There's other types of powerful contributions. You can be more on the financial side of it, like Nordic Sciences is, financing things, or you can be more on the business side of it. And it's very important to have businessmen that create, powerful, profitable businesses. And so yeah, there are different approaches. And my approach, is in some ways the worst, but also the best. It's I mean, the business guys at the end of the day, make these things successful, right? I mean, a technology does not sell itself. You do have to create all the business and marketing and infrastructure to actually impact the world. So sometimes the business people are, at the top, but on the other hand, you don't have anything to do that if you don't have the technology. So, I view myself as having the hard job, but also the job that can actually be most impactful, because you can make something possible that was simply not possible.
For entrepreneurs here located in the Nordics, what would you advise them, deep tech entrepreneurs who wish to sort of expand and grow in US on a global market?
I mean, I think those practical matters. If you want to expand from, a company that was created in the Nordic areas and, do business in the US, I think practically speaking, of course, you need patents in the US, right? You should certainly be sure as you're developing your technology that you're patenting it properly in the US and in the right places for doing business in the US. But, once those sort of mechanical things are taken care of, I think as far as like truly entering US markets, it always comes down, when you're developing deep technologies, you need early adopters and key opinion leaders to sort of adopt and use and promote your solutions. And so it will always come down to those relationships. You have to find the right key opinion leaders, in the US, or who do a lot of activity in the US to test and promote your solution.
Mm-hmm. And now I'm actually going a little bit ahead of myself and then probably back to synthetic biology. But I could ask at this point that what advice would you give deep tech entrepreneurs over here who are sort of scientific founders who starting deep tech companies, and thinking of your experience, and you've been in industry for a very long time, but for a scientific founder, an ex-scientist starting a company in synthetic biology, what advice would you give sort of early on? You mentioned the iteration, how important that is, but anything else that comes to mind?
Yeah, as far as advice to deep tech entrepreneurs, especially say in the Nordic countries, this would be similar maybe to other countries too. But the advice, I think the important advice is you have to pick something you're super passionate about. You have to be extremely passionate, truly emotionally passionate about what you're trying to do. You have to think you're saving the world, because, you're going to have 5 or 10 years of your life wrapped up in this thing. And if you're doing it just for money or something, that's not going to be enough to pull you through all the hard times. There's going to be hard, miserable times when you're almost out of money, or, there's sort of competitors emerging and you feel a huge competitive threat, or, there's internal conflicts with some of your co-founders, employees. I mean, all sorts of headaches. And the only thing that can pull you through those headaches is the belief that you're saving the world. And so you really have to pick something you're truly passionate about. I think that's foundational. You just won't cut it if you don't. And then I think the other thing, especially for scientists, is you have transformative power. Scientists are the true sorcerers and magicians, that's what sorcerers and magicians are scientists. We can, we cast spells and conjure up things that never existed before. And so I think, my message to scientists is you have transformative power. So be sure you're tackling something big, change the world. And so just be sure your idea is big enough. I think Larry Page, one of the co-founders of Google, once said, you should work on something that's basically impossible because then you won't have a lot of competition. And so however big you're thinking, try and think bigger, and try and think of how your special scientific magic could most change the world, and pursue that as the thing you pursue.
That's really excellent advice, and I think that many scientists probably share the sentiments there, and the passion, and also the struggle, some of the struggles that you mentioned, and you have to probably become very comfortable with being uncomfortable as well.
That's exactly right. You live in a state of discomfort. I mean, you have to set the right expectations that life is basically going to be one struggle after another. What I tell people in technology development is like, look, here's how technology development works. It's broken, it doesn't work. And you're trying to make it work. And it's always that over and over again.
Yeah. And then of course, it's an interesting how you communicate that to the other stakeholders and your investors as well, and make sure you're on time over there. But maybe, Barry, I would like to take us back to synthetic biology. There was a quote, there was Mustafa Suleyman, co-founder of DeepMind, in his book, The Coming Wave, he's saying that AI and synthetic biology will redefine society. How do you see these two being really the big areas that are going to shape the world in the future? And also, I'm interested to hear what is the sort of the relationship between AI and synthetic biology that you see?
Yeah, when you look at like what is going to make the biggest impact on mankind over this century, I do think it comes down to synthetic biology and artificial intelligence. And it's interesting, they're really both biological. I mean, the only thing we know about intelligence is biological, right? Trying to find a way to recreate that, but these are both biological things, and I think that speaks to the fact that biology has power that we don't even fathom. Who could have thought that chemistry could turn into humans, or that chemistry could turn into intelligence, which is one of our features. So so I think biology and AI are the biggest sort of factors in this coming century, just in the two areas of the economy. I mean, the things that are going to change the economy, and then the things that are going to change the geopolitical order. The countries that dominate in those things will have a new way to elevate to superpower status. I mean, historically, nuclear weapons gave you superpower status. These things are as powerful as nuclear weapons, in theory. Why is a nuclear weapon powerful? Because it's 1,000,000 times more powerful than what came before. That's why it was special. A nuclear bomb is 1,000,000 times more powerful than a chemical bomb. Well, AI could be millions of times more intelligent than current intelligence in principle. And similarly, synthetic biology, could, basically have it's hard to even put a number on what it could do, because we can literally re-engineer everything, right? And I think when you say what is the nature of the impact that will show up, I mean, synthetic biology is going to impact in two ways. One is sort of radical control over our human biology, which, we mostly worry about our health, but also like our lifespan. And so I think synthetic biology is going to give us radical control over our health. You're going to see a shift from classical drugs and surgery. Drugs are how I get in there and do stuff, and surgery is how I whack at stuff, to biologics in general. Antibodies and engineered immune cells and things like that, which you can put far more intelligence into a biologic than a small molecule drug to go do something important in a disease. And who needs surgery when you can just have cellular remodeling taking care of things? And so, I think that's one huge area where synthetic biology is going to change everything, is in healthcare and wellness and even our lifespan. But then the other big area is manufacturing. In many ways, the world economy runs on manufacturing, and many kinds of manufacturing are going to shift to being made by biology. You can have microbes or something that make the things you want. And so that huge biomanufacturing transition is going to be just the way it actually plays out, is having a massive impact on the economy. I mean, AI is doing what it's doing. I think, and I agree, that the sort of real singularity here is when AI meets synthetic biology. When we truly marry the two together. Why? Because Eric Schmidt, who's the former CEO of Google, but also these days sort of a technology policy leader at the national level, has said, here's how it's going to play out, and here's why AI and biology are so related. Is like, if you think of the revolutions we have had, electronics and mechanical engineering, because of math describes physics. That physics is the thing. Math actually lets us control it, right? Describe it and control it. And he says when you look at biology, it's so complicated. Classical math won't let us describe it properly, but AI will. And so Eric Schmidt liked to say, and I like to give him credit for this because I thought it's a brilliant saying, is what's important here is that sort of AI is to biology as math was to physics. That's why it's so important to bring the two together. In a very practical sense, biology is so complicated. You need AI type capabilities to properly, start making predictions within biology. Again, I think the way it will play out is we will be able to do radically more experiments to get functional data. Be able to make sequences for genes or genomes and see what the thing does and score its functions. And so you'll have sequence to function data at an incredible scale, not just thousands of data points, but billions of data points. And that sequence to function data lets the AI learn how to predict function from sequence. And then you can ask the AI questions like, design a microbe for me that will, make biofuel. And so that's how it will play out. I mean, that's sort of the under the hood. It's going to be creating functional training data and training these AIs to help you do the design work. Some people call this whole thing of the ideal thing then is having a fully automated lab, and especially on a chip. So a fully automated lab thing that an AI can control, because then the AI can go back and forth. The AI can do the experiments it needs to learn and iterate that way. And once you have that exponential iteration, it could very rapidly get to ideal designs for whatever, the ideal antibody to bind some virus, or the ideal microbe to make some chemical. And so, that sort of self-driving lab is probably one of the biggest actual advances. That's how AI and synthetic biology will get married in a very powerful way, is like the self-driving lab.
I think it's so fascinating how we're seeing all the fields sort of fusing together. There's no longer sort of only biology, only engineering, but more and more the interdisciplinarity. And that's something that we also as investors very much look at, that when the inventions, when they are born from this strong interdisciplinarity, but it really sounds like that will even further sort of increase.
Well, one of the problems with being a bio deep tech company, or a deep tech bio company, or whatever, is you fall in a gap in the investors' minds. Investors often see themselves as biotech investors or as tech investors. And, when you're actually marrying those two things, neither one of them know how to value you properly. And so it definitely makes it a little more challenging to get traditional VCs to invest when you are like deep tech bio or bio deep tech.
Hmm. Would you call Avery more of a deep tech or a biotech company?
I think as a deep tech company, I mean, our application is biology, but the things we're developing are all chip devices. You know, we have one chip device that synthesizes DNA. So this is an actual, semiconductor chip made at the same foundries that make the chips in your cell phone. But this one actually carries out lab on chip functionality. It actually synthesizes DNA from primary chemicals and also assembles that DNA into gene-sized things. And we have another chip that can even continue on the assembly to genome-sized things. And so this sort of makes the DNA. And then there's another chip device which actually does the lab work. Like once I've made the DNA, I got to go do lab work with it. I got to put it into a bacteria or put it into a cell-free system and get it expressed and see what it does. And so there's lab work to be done. And this other chip is a so-called droplet microfluidic chip. This has 1 million electrodes on it that can move around droplets of water and carry out all the lab work. Anything you can do
Amazing.
by adding this droplet to this droplet or making something and splitting it up, all that lab work can be done on that chip. And so we are fundamentally a deep tech company. We make chip devices. We have electrical engineers designing chips. We commission foundries to make them. It's just the application is all about making and testing DNA.
Yeah, super. This is very, very interesting. Hey, thank you, Barry, so much for this discussion. Anything final sort of words or reflections you would like to say to innovators in deep tech, just in general? Anything you would like to still sort of are we seeing enough activity on the synthetic biology field? I mean, how to encourage more scientists to really jump into the field?
If we look at the state of synthetic biology, and especially industrial synthetic biology, I mean, synthetic biology has been an academic field for a long time, and that's not the problem. The problem is making it an industrial and commercial field. And so, I think that there's one simple problem there. And it was really highlighted by Craig Venter's work. And Craig Venter is actually a co-founder of our company. He's the chairman of our board. And, he's an inspiration to us, not just because he sequenced the human genome, but then because he turned around and made the first synthetic cell. And but Craig had a very important experience. When he was making that first synthetic cell, he did it at a company. He had a company called Synthetic Genomics. It was one of the pioneering industrial synthetic biology companies. And while his personal passion was making that artificial cell, the company had many contracts to engineer algae to make things. And that they famously had a $600 million contract from Exxon to engineer algae to make biofuel. But they also had many other of these contracts, multi-$100 million contracts to engineer algae to digest plastic from the environment, clean up the environment, pull carbon out of the atmosphere, for decarbonization. And so in total, they had about 30 some contracts to engineer algae to do all kinds of useful things. And the same thing happened in every single one of them. And so bear in mind, this is the most well-funded synthetic biology industrial effort in history. They had $1 billion in funding. These were multi-$100 million contracts. So, they had every resource you could possibly have. They had a fully automated robotic lab and all these things. But the same thing happened in every single project. They could get the algae to sort of do it. Yeah, the algae could make some biofuel, or the algae could, digest some plastic. But they could never make it economically competitive with the classical ways of doing these things. Your biofuel couldn't compete with oil, right? And your algae that eats plastic couldn't compete with just trying to pick up plastic from the environment. And so it was the economics. It was not the scientific possibility. It was the economics. And that means optimization. You get to economics through cost optimization. And so what's missing from synthetic biology is clearly not the will. They had all these contracts. There was a will to do these things. It was not the science. They could make the algae sort of do it. It was the ability to carry out the engineering that is needed to optimize something. And so that's what's missing. And Craig himself said, look, I needed I needed trials on the scale of evolution. When evolution evolves a function, it gets to do billions and trillions of trials. And Craig's opinion was he truly needed trials on that scale to have a powerful way to do these things. And yet you can only afford to do hundreds and thousands of trials because it's so expensive. And so there's a million-fold gap between the number of trials you can do in this trial-based engineering and what you need. And so, that's what's missing in industrial synthetic biology is the ability to make these processes economical. And I think this bites every company that tries to do an application in synbio. It's not that they can't sort of get it to scientifically work, it's that they can't make it economically competitive with what's out there now.
That's really great. I think we will end it here, and thank you so much, Barry, for this interview. Thank you so much.